Welcome. Tonight's show is about osmosis, and we're going to start with a demonstration. What you see in front of you is an array of Thistle tubes, and these Thistle tubes have solutions of varying molarity inside of them. Let me explain. Here's a close-up of the bottom part of the device. You can see that it's a plastic bell. This part over here is what's called dialysis tubing. And that dialysis tubing is sealing the bottom of the tube, but in a very special way, as we'll see. It's filled with a solution of sucrose that's been dyed blue with food coloring. The top is open as well. And so when the entire thing is set up, you jam a plastic straw into the top, you give it some kind of support that keeps it in the midst of the watery beaker, and then you've got your sucrose solution inside with the dialysis tubing on the bottom. Let's see what happens. We're going to run this for just a couple of seconds, but in reality, it took a couple of hours. And here we go. We're in a race, and notice that 1 molar solution has taken off. It's in the lead. It's staying in the lead, followed by the 0.75 molar solution, the 0.5 solution, and the 0.25 solution. The 0 molar solution is hardly doing anything. Well, what we've got is osmosis in action. What is osmosis? Osmosis is simply the diffusion of water. Diffusion of water, that means it's the movement of water from higher concentration to lower concentration. And for this to work, we have to have a selectively permeable membrane. That's what we have over here. And this is a membrane that lets water diffuse through, but not the solute, which in the previous demonstration was sucrose. Over time, water will move from where it's more concentrated to where it's less concentrated, and that'll change the level of water on each side of the membrane. That has huge consequences for cells, both animal cells and plant cells, which are hugely affected by osmosis. It shows up in some very important labs that are done in the AP Bio curriculum and in other curricula as well, including the dialysis bag lab, the potato core lab, and we'll talk about a diffusion lab. That's what's happening tonight on the AP Bio show. On the show tonight, we're also going to have a song of the week, the Osmosis song, a weekly quiz, and there'll be tips for learning the greatest subject of all, biology. A little bit about your host. I am Glenn Wokenfeld, also known as Mr. W to my students. And this year, right now, you're my students because I'm a recently retired AP Bio teacher and I've taken my teaching online. My mission is to help you succeed in your AP, IB, or college biology course. One of the main ways that I do that is through my website and my app. My website is called learn-biology.com, approved by the College Board, used by hundreds of teachers, thousands of students to help them get 4s and 5s on the AP Bio exam and A's in their biology course. I'll tell you more about that later. Let's start with our lesson of the week. Tonicity and osmoregulation, that's the College Board's name for it. You might want to call it something simpler like osmosis and its consequences. Let's start by talking about what osmosis is. Osmosis, again, is the diffusion of water. Water is going to move from higher concentration to lower concentration. What does that mean? If you look at these solutions, like, for example, this one over here, this one has very little solute inside of it. It's mostly water. Let's say it's 98% water by weight, 2% solute. This has more solute inside of it. That means that the percentage of water is going to be lower. So water's in higher concentration on this side and lower concentration on this side. That's going to cause water to move from where it's more concentrated to less concentrated. There are some special terms that we use to describe this movement of water. Water flows down its concentration gradient from hypotonic to hypertonic. hypotonic. What does that mean? Hypotonic is a solution that has relatively more water. So this is the hypotonic side. It has relatively less solute. Hypertonic means that it has relatively less water and relatively more solute. And notice how I keep on using the word relative over and over again. There's nothing that's absolutely hypotonic or hypertonic. This is always about two solutions in relationship to one another across a space, or more commonly in biology, across a membrane. This is something that needs to be memorized. Water always diffuses from hypotonic to hypertonic across a membrane or from one area to another. Water flows from hypotonic to hypertonic. Here's an example. It's called a U-tube. This is a device that you can screw apart and then you can put in a membrane. The membrane that we're going to use is going to be selectively permeable. And in this case, it's permeable to water, but not to the solute. We're adding sugar to one side of it. And what that does is it makes this side hypertonic relative to this side, which is hypotonic. Because water always flows from hypotonic to hypertonic, we can see this side of the U-tube rise and this side of the U-tube fall. It's all about osmosis, and just remember, water always flows from hypotonic to hypertonic. Now, this isn't happening by magic, and people will often say things like, water likes to move from the hypotonic side to the hypertonic side, but here we are studying science. We can lift the veil a little bit and look underneath. When you add solute, you're adding, in this case, sugar. And sucrose has all of these hydroxyl groups on the outside. The water molecules are in random motion. The sucrose molecules are in random motion too, but they're stuck on this side of the U-tube. Why? They're stuck because the membrane is not permeable to sucrose. So these water molecules are going to randomly move across, and when they move, they're going to interact with the sucrose molecules, and they're going to hang out with them. Why? Because they're going to form hydrogen bonds with all of these hydroxyl groups. So the water molecules get kind of stuck on this side in their interaction with the sucrose, which randomly will move more molecules from this side to the other side. That's what's happening on a molecular level, and that's how you should think about it. Here's another fabulous example. A student places a gummy bear in a bowl of water overnight. The next day, the gummy bear has expanded. Explain what's going on. Well, the gummy bear is mostly made of sugar. The water in the bowl is, no surprise, 100% water, or nearly so. Water is going to flow from the hypotonic solution in the bowl, it's mostly water, into the hypertonic gummy bear. And as water moves through into the gummy bear, it's mostly going to stay where it is. That's going to cause the gummy bear to expand. Again, I have a song about this called Osmosis. I'm going to play it for you later in this show. Here's the Thistle tube demonstration that we started with. What's happening? This solution is the most hypertonic. It's 1.0 molar. Water's going from hypotonic to hypertonic, but the gradient is higher over here in the most hypertonic solution. So the water moves more quickly into the 1.0 molar solution than it does into the other solutions. Let's go on. Osmosis in plants. Use the principles of osmosis to explain each of the images. For those of you in the listening audience, I have 3 diagrams. The first one shows a kind of shriveled up plant cell, and water is leaving that cell. In the middle diagram, water's leaving and entering at the same rate. And in the cell on the right, water is entering the cell, and you can see how the vacuole is much bigger in the cell on the right than it is in the cell in the middle, than it is in the cell on the left. What's going on? In the cell on the left, we have a situation where the cell is hypotonic to its environment. In other words, the cytoplasm had less solute dissolved in it than the solution outside. As a result, water is going to flow from the hypotonic cell to the hypertonic solution outside the cell. Water will leave the cell. The process is called plasmolysis. The membrane will peel away from the wall. The vacuole, which is the primary water storage organ, will shrink. In the middle cell, the cell is isotonic to the environment. That's a new term. Isotonic means that the amount of dissolved solute is the same in both solutions, or you could say the percentage of water is the same in both solutions. So water will enter and leave at the same rate. There'll be an equilibrium. In the cell on the right, the cell is hypertonic to its environment. That's what happens when you water your plants. You create a situation where outside the cell is hypotonic to the interior of the cell. Water is going to move into the cells of the plant as a whole, and that is going to lead the vacuole to expand. It's going to push the entire cell against the cell wall. That's called turgor pressure. It makes plants full, firm, happy. That's why you should water your plants. So explain this. This is a water plant. It's called Elodea. It's frequently used in biology labs because the leaves of this plant are extraordinarily thin, only a few cells, and you can just take off a leaf and lay it on a microscope and take a look. Number one in this diagram is a chloroplast. Number two over here is the wall. Here's Elodea in its normal environment, and here's Elodea placed in salt water. And again, you can do this in the laboratory easily enough. The salt in the solution on the right side makes the cells hypotonic to their environment, and water will leave the cell. The membrane will peel away from the wall. That's what you see over here. And again, that's plasmolysis. That's this process of the membrane shrinking away from the cell wall. And it's actually an easy way to visualize the cell membrane of plant cells, which otherwise is going to be pressed against the cell wall and difficult to see. Osmosis in animal cells. Use the principles of osmosis to explain each of the images below. On the left, the cell is hypotonic to its environment. In other words, there is more solute outside of the cell than there is in the cell's cytoplasm. As a result, water is going to flow from the hypotonic cells into the hypertonic environment, and the cell will shrink and shrivel up. In the middle, the cell is isotonic to its environment, so water will enter and leave the cells at the same rate. And in the right side, the cell is hypertonic to its environment. There's a higher concentration of solute inside the cells than there is outside the cells. That's what would happen if you just put cells in water. Water will flow into the cell, and because these are animal cells, they don't have a wall, they'll expand and expand until the membrane ruptures. Here's a cool thing. Paramecia are these lovely single-celled eukaryotes. You can easily see them with a microscope, and they have an organelle that's called a contractile vacuole. The contractile vacuole is an evolutionary solution to a problem faced by freshwater protists, which is that they are living in a lake or a stream, and the amount of solute in their cytoplasm makes them hypertonic to their watery environment. And as a result, water is moving into these cells by osmosis. Now, the paramecium happens to have a structure that's called a pellicle that helps maintain some of this pressure, but the contractile vacuole also plays a role in maintaining the correct balance of water in the cell. Here, you can see the contractile vacuole very much expanded, and that sets us up to watch a video of the contractile vacuole at work. The contractile vacuole is going to fill with water and then contract to expel water from the cell. This is a form of osmoregulation. It's regulating the internal water balance of an organism. You can actually experimentally manipulate this in a lab, and it's fun and easy to do. You can change the molarity of the solution that the paramecium is in. When it's in pure water, distilled water, for example, the contractile vacuole will be contracting at its maximum rate. As the molarity of the solution increases, the contractile vacuole will be able to contract at a slower and slower rate until at some point the paramecia will be isotonic to its environment, and then the contractile vacuole won't have to contract at all. Get a microscope, get some sugar, get some paramecia. You can test it out for yourself. See if you can figure this one out. White blood cells are placed in solution. Based on what happens, determine whether the solution was hypotonic, hypertonic, or isotonic to the cells, and explain. Let's watch. Okay, let's see what's happening to those cells. Watching them. Holy cow! Look at these poor cells. Well, what had to happen here? The cells increase in size and then they burst. The cells must have been in a hypotonic solution. Water flowed from the solution into the cell, causing it to expand in size. And because this is an animal cell, it's a white blood cell, there's no wall, and ultimately the cell bursts from osmotic pressure. We're now going to move on to some osmoregulatory features of plants. Stomata are used for osmoregulation in plants to describe their structure. Here's a drawing of a leaf of a plant, and here's a cross section of that leaf, and there's many layers of cells. The top and the bottom layers are both referred to as epidermis, that's the outer covering, and the epidermis is covered by a waxy cuticle, and that prevents water loss from these leaves, which are exposed to the wind and the sun, so on and so forth. Now, the plant, and the leaf in particular, needs to exchange gases with the environment. And there are these pores on the bottom that are referred to individually as a stoma and collectively as stomata. So here you can see them on the bottom, and here you can see what they look like when they're viewed from one side. And again, this is another typical lab in an AP Bio class where you investigate leaves to look at the stomata. And these two cells, here's one below, here's one above, they're called guard cells. How do these stomata work? When there's sufficient water, the guard cells will buckle outwards. This creates a pore, that's the stoma, that allows carbon dioxide to enter the leaf for photosynthesis, but that creates a problem because it also allows water vapor to escape. And that's a normal thing for plants, but it's obviously not healthy for plants to lose too much water. They'll wilt. And so plants control that, and they control that at a cellular level. The stomata close in response to a variety of environmental clues. One is light, but another one is water stress. How are these guard cells regulated in order to open and close? When water is available, the adjacent cells, so not the guard cells themselves, but this cell over here, this cell over here, or this one over here, this one over here, they pump potassium ions into the guard cells. Here you see potassium that's getting pumped in. Water follows by osmosis, because essentially what happens is that these cells become hypertonic. Water flows from the hypotonic cells adjacent into the hypertonic guard cells. And there's an array of fibers that force the guard cells to open up like this. That creates the stoma over here at number 2. When water is scarce, the pumping stops, the potassium ions flow out by diffusion, and water follows, and that causes the stomata to close. Understanding stomata is the key to understanding the transpiration lab, which I hope you do. It's widely done in the AP Bio curriculum, but it's a way to measure water loss from plants Water gets lost as water moves from the soil into the roots, up the stem, and out the leaves. And that process is called transpiration. It's incredibly important, and in fact, it's responsible for things like the amount of rain that happens over forests. And you can simulate that very simply with a plastic bag and a tiny little plant like this broccoli plant. I'll talk about that later in the year. Do you want to learn more? If you want to learn more, then just go to learn-biology.com and complete the tutorials about osmosis. I understand that this is tough stuff, and the stuff that I just explained, I explained rather quickly. You need to interact to learn biology. And with learn-biology.com, you will get the skills and the confidence that you need to ace your biology class and the AP Bio exam. Learn-biology.com is 100% aligned with the College Board's AP Bio standards, and it's approved by the College Board as a textbook alternative. Learn-biology.com has interactive tutorials about every topic in the curriculum, and we have readings, videos, flashcards, quizzes that teach you everything you need to know about every topic in AP Bio. With learn-biology.com, you will interact, and you'll interact through multiple choice questions, click and drag diagrams, identify the part questions, free response questions. We have games. You get positive feedback for your correct responses, and you get hints and repetition for incorrect responses. So by the time you are done with the tutorial, you know the stuff. We also have student learning guides that tell students exactly what to do as they move through the tutorials. And we have an AP Bio review system that tells you everything that you need to crush it on the AP Bio exam. If you're a teacher, you'll be happy to know that we provide student performance monitoring, a kind of grade book, so you can track your students' performance. We have free trials available for teachers and for students. Go to learn-biology.com, learn more, and prepare to crush it in your biology course. Let's move on to the topic of water potential. Water potential is a quantitative way to understand the material that we've just learned about in relationship to osmosis. And water potential, which is represented by this Greek letter psi, is a measurement of water's tendency to move from where it is to where it's not. Adding solute, like pouring sugar into one side of this U-tube, decreases water potential. Pressure increases water potential. So you would generate pressure by pushing on the arm of this syringe over here. The rule to remember for water potential is this. Water flows from areas of high water potential to areas of low water potential. In the same way that you want to memorize that water moves from hypotonic to hypertonic across a selectively permeable membrane, water will move from higher water potential to lower water potential. Here's the formula. The formula can be more complex than this, but for AP Bio, all you need to know is that water potential equals solute potential plus pressure potential. Again, the Greek letter psi represents water potential. Psi sub s is solute potential. Adding solute decreases the water potential of the body of water. Psi sub p is pressure potential. Adding pressure, like pressing on the plunger of a syringe, increases water potential. With that in mind, using water potential, explain the diagram below. When we add sugar to this side of the U-tube, what happens is this. The pressure potential is 0. There's no pressing on either side of this U-tube. But adding the solute lowered the water potential. It made it to negative 0.23 megapascals. That's the measurement for pressure. That's something that really comes out of physics. And all you need to know is that megapascals is a measurement of pressure. So the water potential over here is 0. There's no solute, there's no pressure. And water will move from higher water potential 0 to negative 2.3. And as a result, the level on the left side will rise. We explained this before with terms like hypotonic and hypertonic. And notice that I did the same thing with water potential, but now we've quantified this process. Using the concept of water potential, explain what's happening in the diagram below. This is some tissue. It could be potato tissue, which is typically done in an AP Bio lab. So what's going on here? The cell is in water. In this situation, there's no pressure. So the pressure potential is 0, but there's a lot of stuff dissolved in cells. So the solute potential is -1 megapascal. So what's your total water potential? It's -1.0 megapascals. Now, what'll happen is after a certain period of time is water will flow from 0 to -0.1. But at a certain point, there'll be an equilibrium reached. Well, what will that be about? The water flowing into the cell will generate what's called turgor pressure. That'll rise, that'll rise, that'll rise until it gets to 1.0. And at 1.0, basically we have 1.0 plus minus 1.0 equals 0. So we've created an equilibrium. You can pause this slide and read all the bullet points, but I just explained it, so I'm gonna move on. Let's have a special topic, sidewalks and osmosis. You might think that osmosis is something that only affects you in the laboratory. Think again. If you go for a walk along your neighborhood and you see something like this with tree roots and then an uplifted sidewalk, think to yourself about what force caused a concrete sidewalk to break and get lifted up. And the answer, of course, is osmosis caused that. Plants don't have muscles. Plants don't operate jackhammers. Plants don't have levers. But what happens is that the cells in the root absorb water by osmosis. That caused the cells to expand. As the root grows, that process continues and continues and continues, generating sufficient force to crack concrete. That's how powerful osmosis can be. Let's see if there's another special topic, and there is. We're going to talk about the diffusion and osmosis labs that you're probably going to be doing in this part of the AP Bio curriculum. All of these labs use dialysis tubing. I've talked about it before. Here's some dialysis tubing that's just laid flat. This is what it looks like when it's dry. The main thing is that dialysis tubing is selectively permeable. The dialysis tubing that we'll be using in labs is so impermeable to sucrose, which is a disaccharide, and larger molecules like polysaccharides and so on and so forth. So lab number 1, you do this. You have sucrose solution that's in dialysis bags. So these bags are tied off after being filled with various amounts of sucrose solution. The cup into which they're placed has distilled water, but for those of you biology teachers out there or homeschoolers who want to do this lab and economize, it'll work just fine with tap water. The bags have sucrose solution, and what you do is you place all of these bags with varying molarity of sucrose solution into the water at the same time. You let them sit for about 30 minutes. You place them all in the water and predict what'll happen. When you graph this data, here's what you'll find. You'll find that the 1.0 molar sucrose will have gained the most mass. It'll have the highest percent change, where the 0 molar sucrose will really not change at all. This might just be within the margin of error. And as the bags become more hypertonic, the amount of mass that they gain increases. And it's perfectly correct to draw a trend line, because what you're doing is you're measuring one value. It's a continuous value. And you could make up a sentence like this, like, as the molarity increases, the percent mass gain also increases. It's a continuous value, and that's why we graph it like this. So that's dialysis bags with sucrose. That's what happens. Another variation on this lab takes potato cores in solutions of varying molarity. So you take potatoes, you use what's called a potato core. It's a cute little device, and you wind up with these little potato cores. And then what you do is you mass them, and then you put them in solutions of varying molarity. So here's 1.0 molar sucrose, here's 0.66, here's 0.33, and here's 0 molar sucrose, which would just be water. So predict what will happen to the mass of the potato cores over time. And what you find is that when you calculate percent gain, what you'll see is that some of the potato cores will have gained mass, and some of the potato cores will have lost mass. What's that all about? At 1 molar sucrose, the potato tissue is hypotonic to that 1.0 molar solution. So water will flow from the potato tissue into the solution in the beaker, and as it does, those potato cores will lose mass. The 0.66 molar solution looks like that one is also hypotonic. Those cells are hypotonic to the solution, and therefore that molarity will cause these cells, these potato cores, to lose mass. But at 0.33, this solution must be hypotonic to the cells in the potato tissue, and water will move from the solution into the potato cores, causing them to gain mass. And when you have these potato cores in pure water, they'll gain even more mass. And there's something very interesting. When you draw the trend line, you'll notice that at a certain point, the trend line will cross 0% change. What is that? It's really cool. And if you can think of this yourself, I'll give you a moment to do that, then you're really understanding this very well. But at that molarity, you've determined the tonicity of the potato core tissue. At this molarity over here, that's where the solution is isotonic to the potato core tissue. So who knows exactly where that would be? Maybe it's about 0.37 or something like that. But that is a way that you can figure out the molarity of the tissue. Is that cool or what? Now, there's one more lab that is part of this series. It's not directly about osmosis, but it is about diffusion. And here's what you do. You take water, you add iodine, potassium iodide solution to it. So that's your starting solution. That's why it has this nice red color. Into that, you add a bag that's full of fructose starch solution. So, starch, you know what that is, a polysaccharide. Fructose is a monosaccharide. You mix together. This can also be glucose. Any monosaccharide will do the trick. And then you take some of this solution and you pour it off into the test tube to preserve its initial color. If you give this a couple of hours, then you'll find that the fructose starch solution is initially white. And then overnight, it'll turn purple. You need to know a little bit about the chemistry here. When iodine interacts with starch, it turns purple. So what that means is that the iodine had to somehow diffuse through the bag and interact with the starch, but it's inside a bag. You'll also notice that, in addition, this solution has become a lot lighter than it was before. So we have all these changes. And yet another thing, which I'm not really demonstrating over here, if you test this solution afterwards for the presence of fructose or glucose, you do a monosaccharide test, you'll find that there is some glucose or fructose present in the beaker, in the cup that you're doing this experiment with. What's going on? How does this happen? It ain't magic. It's all about diffusion. Here's a table that summarizes what happens. The initial color of the solution of the bag was white. In the end, it's black or purple. The color of the solution in the cup was darker red. At the end, it's lighter red or even clear. Is fructose present in the cup at the start? Nope. Is it present there at the end? Yes. Was iodine present in the bag at the start? Nope. Was it there at the end? Yes. And here are two diagrams that demonstrate what's going on. How do we explain this? What I want you to keep in mind is that there are three substances. There are iodine ions, there's fructose, which is a monosaccharide, and then there's starch. Here's before, here's after. Thinking biologically means thinking on a molecular level. So let's do that. On a molecular level, the dialysis membrane has tiny pores that some molecules can pass through, but not others. That's how it works. Starch, which is a polysaccharide, is way too big to pass through these pores. Fructose, a monosaccharide, is much smaller, small enough to be able to pass through. And what's true of fructose, a monosaccharide composed of 24 atoms, is also true of iodine, a single ion. I like to imagine myself being the size of a molecule of starch. On that scale, the dialysis tubing would be like a chain link fence, too big for a starchy molecule my size to pass through. On that scale, the fructose is like a ping pong ball, which can easily pass through a chain link fence, and the iodine is like a marble, which can pass through the fence even more easily. Why is this important? What we've done in this lab is to demonstrate the properties of a selectively permeable boundary. And while the structure of a cell membrane is completely different from this dialysis tubing, the membrane is also a selectively permeable boundary. It lets some things pass through, but not others. And that selective permeability is really the basis of all living systems. The song of the week is a song that's on the Science Music Videos channel. It's called Osmosis. I want to do a shout out to my wife, Nancy, who helped me do the little stop motion animation that you will see at the start of the video. We're going to go here.
I put this yummy gummy through a science experiment that models what happens to a cell in an environment that's more watery than the cell is inside. And what I saw came as no surprise. My gummy expanded, quintupled in weight. My gummy expanded and increased so great. So listen up as Mr. W explains this. It's all about osmosis. Osmosis is the diffusion of water across a membrane or a water permeable border, diffusing like all molecules in fluid situations from higher to lower concentration. Osmosis, H2O diffusing. Osmosis, see the water oozing. Hypotonic to hypertonic flow. Osmosis, let's go. Let's talk about our gummy and osmotic terms. Hypotonic, hypertonic, isotonic are the words you can use to discuss water's watery diffusion. Learn them well to avoid confusion. Hypotonic means higher water concentration and relatively lower solute concentration. Hypertonic is the opposite, percent of water's less, with more solute dissolved inside, as you might guess. So let's take these terms and apply them to our gummy bear. I'm looking at the package and seeing what it says here. Each bear is mostly sugar with some other stuff mixed in, and holding it together is the protein gelatin. So when you put a gummy in a cup of H2O, it's readily apparent that the gummy is so hypertonic to the water that it's in, and the water's hypotonic to our little gummy friend. And so through osmosis, the water will diffuse into the gummy, which soon will look so huge. The mass it gains is water, which will infiltrate the gummy, which one day later is looking pretty funny. Osmosis, H2O diffusing. Osmosis, see the water oozing. Hypotonic to hypertonic flow. Osmosis, let's go. Next I took this freshwater plant, name is Elodia. It lives in lakes and ponds, never in the salty sea. In freshwater, Elodia cells are full and firm. It's all about osmosis, as we're gonna confirm. See, the cells are hypertonic to their watery exterior, so water will diffuse into the cell's interior, expanding the membrane, pushing it against the wall. In fact, you can't see the membrane at all. But add some salty water so the outside's hypertonic, and water leaves the hypotonic cells, it's so osmotic. Membrane leaves the wall, the cells inside the scrunch, with the chloroplast are stuck together in a bunch. That's why at the grocery store they always have that mister. The water on the veggies helps to keep them crisper. The droplets on the veggies are a hypotonic brew. Osmosis moves the water in, the veggies look brand new. Dried fruit and beef jerky preservation is osmotic. Their low water content makes the dry food hypertonic. So any germs and molds that fall upon them lose their water and die, and don't contaminate the dry food in the larder. Osmosis. H2O diffusing. Osmosis. See the water oozing. Hypotonic to hypertonic flow. Osmosis. Let's go. Animals, cells and situations hypertonic, lose their water, shrink and shrivel. Looks so sick, but an animal cell in hypotonic abode gains water and expands, eventually explodes. Cause animal cells lack a wall, of course, so nothing pushes back at the osmotic force. Water flows from hypotonic to the hypertonic cell, which bursts cause the membrane can't stop the swell. If you want to keep a frog heart outside a body beating, an isotonic fluid is what you'll be needing. Water concentration same on each side of the cell, no gain or loss of water, the cells feel so swell. And a paramecium, which lives in ponds and lakes, constantly fights osmotic water uptake. The contractile vacuoles a pumping adaptation to deal with this osmotic situation. Osmosis. H2O diffusing. Osmosis. See the water oozing. Hypotonic to hypertonic flow. Osmosis. Let's go.
Thank you so much for taking the time to watch Osmosis. I'd like to ask you to do a couple of things. Please subscribe to my channel, mark it as a favorite, like it, leave comments. I love it when people leave comments. It means a lot to me. Please share by tweeting about my videos, posting them on Facebook. I'm also selling MP3s, and I just launched a website, www.SCIENCEMUSICVIDEOS.com, getting more curriculum materials there all the time. And thanks a lot. Look for more videos. Really appreciate you joining me to watch Osmosis. Thanks. And we've done our song of the week, so we are on to our weekly quiz on learn-biology.com. Now we're on learn-biology.com. We're going to jump down to our Unit 2 Cumulative Multiple Choice Quiz 1. Let's throw in questions about membrane transport and membrane permeability. First question. A scientist has constructed an artificial cell membrane that consists solely of a phospholipid bilayer. A portion of this membrane is shown below. The image, for those of you who are listening, just shows a phospholipid bilayer. Which of the following substances would diffuse most rapidly through this artificial membrane? The first choice is glycogen, a polysaccharide. The second choice is glycine, an amino acid. The third choice is fructose, a monosaccharide. The fourth choice is Ca++, the calcium ion. And the last choice is O2, an oxygen molecule. Think about it. What could most easily diffuse through this phospholipid bilayer? Everybody's thinking. And the answer is oxygen. And the feedback that you're getting from our quizzes on learn-biology.com is nicely done. Small nonpolar molecules like oxygen, along with other such molecules like CO2 and N2 nitrogen, can diffuse through phospholipid bilayers. Next question. Estrogen is a steroid hormone. Its structural formula is shown below. It's showing the structural formula of estrogen, which is a bunch of fused carbon rings with hydroxyl groups on both ends. Which of the pathways would estrogen diffuse through a cell's membrane? And again, for those of you in the listening audience, 1 shows a substance moving directly through the phospholipid bilayer. 2 shows a substance that is moving through a protein channel. And 3 shows a substance that is moving from low concentration to high concentration, again through a protein channel, and expenditure of ATP is required. So which of the pathways would be the right one for estrogen? 1, 2, or 3? Now, just to demonstrate something, I'm going to show you what happens on learn-biology.com if you were to choose a wrong answer. Let's say you chose 2. Here's what the program is telling you. It says no. Because estrogen is a non-polar lipid, it can diffuse directly through the phospholipid bilayer. Which number shows this type of a diffusion? I'm looking at the diagram and I'm seeing, oh, it's number 1. I'm going to remember that when that question cycles around next time. What's the next question? Amoebas are unicellular protists that are found in aquatic environments. The diagram below shows the same cell, the same amoeba cell, in 3 different environments. One of these environments, X, is the amoeba's natural habitat. Environments Y and Z have been experimentally manipulated. So what we see are 3 cells. In X, I don't know how to describe this, but it's an amoeba that is of a certain size. In environment Y, the size of the cell is much smaller, and in environment Z, the size of the cell is much bigger. So X, normal size, Y, the cell is smaller, Z, the cell is bigger. Based on the information above, environment Y must be blank to the amoeba's natural environment. And your choices are 1, hypotonic, 2, isotonic, 3, hypertonic, and 4, mesotonic. Here's how I'm thinking about this. In environment Y, the cell has decreased in size. The way that would happen is that water would have to leave the cell, and therefore the environment must be hypertonic to the amoeba's natural environment. I'm going to click that choice, the 3rd choice, and excellent. Water obviously left the amoeba, causing it to shrivel up. Because water always flows from hypotonic to hypertonic, that would only happen if the cell were in a hypertonic environment. Next question. A part found in plant cells but not animal cells that allows plant cells to thrive in hypotonic environment are aquaporins. I didn't talk about aquaporins. Aquaporins are specialized pores that allow water to more easily pass through the plasma membrane. The second choice is chloroplasts. The third choice is Golgi bodies. The fourth choice is the cell wall. And the fourth choice is the rough endoplasmic reticulum. Do we have any idea what this is? Think about it. Now, I'm again going to demonstrate what happens if you make a wrong choice. I'm going to choose those aquaporins, and it says no. Aquaporins are membrane channels that facilitate the flow of water across the membrane. The challenge of a cell in a hypotonic environment is to avoid bursting as water flows from the hypotonic environment into the hypertonic cell, as shown in the red blood cells on the right side of the diagram below. What plant cell structure could prevent a cell from bursting due to osmotic pressure? Well, we'll see that question again. Next question. The permeability coefficient measures the ease with which a molecule passes through a cell membrane. The graph below displays the permeability coefficients of 6 different molecules with various solubilities in oil. Which of the following statements is supported by the data in the graph? For those of you who are in the listening audience, the x-axis is showing solubility in oil in arbitrary units. The range is from 0.001 to 0.1, and the y-axis is showing the permeability coefficient, which goes from 0.01 up through 10. Water is at a permeability coefficient of, I don't know, about 5, and its solubility in oil is very low. Urea has very low permeability coefficient and very low solubility in oil. Ethylene glycol has relatively low solubility in oil and a sort of medium level permeability coefficient. Alcohol has a high permeability coefficient, the highest, and a somewhat high solubility in oil. Codeine is lower than alcohol, but it has the highest solubility in oil, and diethylurea has a sort of medium level permeability coefficient level and solubility in oil. Which of the following statements is supported by the data in the graph? The first choice, alcohol is less lipid soluble than water. The second choice, excess water is removed from a cell faster than urea. 3, diethylurea can more easily move through a cell membrane than alcohol. And 4, ethylene glycol can more easily move through a cell membrane than codeine. But I'm thinking, which is supported by the data in the graph? Alcohol is less lipid soluble than water. No, well, I can see alcohol is obviously more soluble than water, so we're eliminating this choice. Excess water is removed from a cell faster than urea. Well, water has a much higher permeability coefficient than urea, so that sounds pretty good. Let's look at the third choice. Diethylurea can more easily move through a cell membrane than alcohol. Diethylurea has a lower permeability coefficient, and it also is a little bit less soluble in oil, so I think that's wrong. And ethylene glycol can more easily move through a cell membrane than codeine. No, codeine has a higher permeability coefficient than ethylene glycol. So I think I'm going to go with choice 2. Excellent. Water has a higher permeability coefficient than urea. Therefore, water would more easily move through the cell membrane. Next question. Here we have a pretty complex diagram that I'm going to describe to you. The diagram below illustrates a process related to glucose homeostasis in liver cells. So what we have is a schematic diagram of a cell. On the left side, there's an insulin receptor and an insulin and dots that represent insulin. Below the insulin receptor, there's a kind of signal transduction system. That signal transduction system goes into some of the internal membranes of the cell. And what we see budding off are these things called GLUT4 vesicles, which then fuse with the membrane, which then go to the cell membrane, and they allow glucose to enter the cell. The glucose is entering the cells by which process? The first is simple diffusion. And I guess what you need to know for this is that glucose is entering the cell. It's going from higher concentration to lower concentration, and it's going through this GLUT4 vesicle. So the glucose is entering the cells by simple diffusion is the first choice. Receptor-mediated endocytosis, that's the second choice. Active transport is the third choice, and facilitated diffusion is the fourth choice. What do you think it is? I think that looking at this diagram, I'm seeing glucose entering into a cell through a protein channel, and that says to me, facilitated diffusion. Bam! This was facilitated diffusion, which involves diffusion through a protein channel, as illustrated in number 2 below. And this is a diagram that compares simple diffusion with facilitated diffusion with active transport. Next question. Oh, here's the one that we got wrong before, because questions repeat. Estrogen is a steroid hormone. Its structural formula is shown below. Estrogen is a bunch of fused carbon rings, does have 2 hydroxyl groups on the outside. By which of the pathways below would estrogen diffuse through a cell's membrane? So what I'm thinking is that estrogen as a steroid is mostly non-polar. Non-polar things, I remember from the previous question, are capable of diffusing through the lipid bilayer through the process of simple diffusion. That's number 1. I'm going to click number 1 this time. Great job. Because estrogen is a non-polar lipid, it can simply diffuse the phospholipid bilayer, as shown in number 1 above. Here's another question. A part found in plant cells but not animal cells that allow plant cells to thrive in hypotonic environments is R. This is a repeat of a question that we got wrong before as well. The choices are aquaporins, chloroplasts, Golgi bodies, the cell wall, the rough endoplasmic reticulum. I remember from the hint that I previously received that the cell wall acts as a kind of pressure vessel, and that plant cells are happy when they're in a hypotonic environment because that pushes the membrane against the wall, it expands the central vacuole, it keeps the cell full and firm. And that's correct. Excellent. The cell wall is what plant cells use to keep from bursting from osmotic pressure in a hypotonic environment, as shown in the cell on the right below. I'm clicking for a summary report. Congratulations, you're done. In finishing the 6-question set, you entered 2 incorrect answers. These are the topics that you answered incorrectly. And I'm told the 2 topics, one was about membrane transport, one was about tonicity and osmoregulation. That's the kind of feedback that you get on learn-biology.com. Your plan for this week. And your plan for this week is very straightforward. Your plan is to go to learn-biology.com, then go to courses, then go to AP Bio, then go to Unit 2, Cell Structure and Function. We're at the end of Unit 2. A lot of teachers are already transitioning to Unit 3, which we're going to start next week. You're going to review osmosis and water potential, which is the material that we just covered in this video, and then go to the unit review page that we just used for the quiz. Do the 2 multiple choice quizzes, do the flashcards, do the FRQ quiz, do the game, and you will know about cells so well you won't believe it. You'll feel great and so confident. And when you walk into that unit test, you're going to be ready to crush it. So I'm counting on you to do that. We are going to now move on to the next video in this series. Please subscribe to the Science Music Videos channel. Watch this next video. Thank you so much for spending time with me here tonight.